So, Dr. White, first of all, let me thank you so much for being available. I have been stalking your work for some time now. And in preparation for the current exhibition in our Portland, Maine location, which is called Seed to Soul, I have been researching really largely roots and root hairs and soil, and then also the really fascinating things that take place with mycorrhiza and rhizophagy exchange. And of course, every time I search rhizophagy exchange, you come up and this fascinating presentation around cannabis and all of your research. So if you wouldn't mind taking a few minutes to introduce yourself and your background, that would be great.
Thank you, April. I am James White, a professor at Rutgers University in New Jersey in the Department of Plant Biology, and my research focuses on really endophytic microbes in plants, and these are microbes, fungi, generally fungi and bacteria that enter plant tissues. And I've been doing this kind of work for over 40 years now, probably close to 40, 44 years, something like that now. So it's a long time I've been working in this area, and very, very gradually, we got to an understanding of what's happening inside plants. And yeah, I'm a pretty old guy. I can tell you that already. Pretty old guy. But I love what I'm doing, and is that enough information about me,
Absolutely.
April? Okay.
Yeah.
Okay.
And so the way that the exhibition works is I'm trying to take people from the seed all the way to when THC starts to interact with your body and other cannabinoids. So it's called Seed to Soul because we're trying to track it. And the way that I diced it up is one of the exhibits talks about soil, and then another talks about roots. And so I've sort of artificially separated these two things, even though they're really combined. And so I'd like to start with roots, if at all possible. And I want to really just get you to describe, an overview of roots. I know that could take a long time, but just a few fascinating things about the cannabis roots themselves, and then explain mycorrhiza and how that really adds to what the roots are capable of.
Yeah. So roots, right? What are roots? We all have some concept of what roots are, right? They're these little plant extensions that go into the soil and ramify and get nutrients, right? But actually, the roots of plants are analogous to the intestines of animals, right? They are the structures whereby plants interface with the soil and interface with soil microbiology and obtain the nutrients that they need. I mean, it's basically like a stomach in the soil, an inside-out stomach, if you will, that goes into the soil. And around we learned that there are these symbiotic interactions that happen in roots and on roots, and mycorrhizae. These are fungi that actually have evolved the capacity to interact with roots, to get nutrients from roots, also provide nutrients to plants from the soil. And so these are specific groups of fungi, the mycorrhizae, basically myco, fungus, rhizae, root, fungus root, right? That establish a symbiosis with plant roots and provide nutrients and obtain nutrients from the plant. And that was one of the first symbiotic interactions that we understood were happening in roots, or these mycorrhizae, because people could see them in there.
in addition to, being this sort of microbiome, is that like similar to our own microbiome in our gut? These mycorrhiza are going out there and helping digest and pull in nutrients. In addition to that, is there communication taking place between separate plants or separate trees through these mycorrhizal connections that connect those roots?
Well, we know the concept of the so-called wood wide web, right? Where for example, trees in natural communities, like forest trees, will have fungi that will mycorrhizae that will grow on one plant, and then mycelium will go out from that one plant and connect to other plants. So you have this network, right? And there is, without doubt, there is sharing of nutrients between communities, between individuals in a community through mycorrhizae. And we know that, for example, that larger trees will connect to smaller trees, and the smaller trees will then have a boost up, a leg up, because they're being fed essentially by nutrients coming from the larger trees. So there is definitely that. Sometimes we carry the idea too far in that, there's this big communication network, and there is, but how far it goes is really the frontier that we're not sure, right? How much of that actual communication is happening. That's where we're learning more and more.
It's fascinating. I mean, I'd love to believe that the parent plants are nurturing the baby plants, right? As a mother, that is really enticing to me to think about.
And they are. They really are.
Oh, that's fantastic.
Yeah.
I love that dynamic. Is there any part of the mycorrhiza relationship with the roots that you don't think is covered enough in the research, or something that people might not know even if they've dug around for about this topic?
Well, we're learning all the time new things. There are lots of people looking at mycorrhizae. It's very important symbiotic interaction between plants and fungi. So people are really digging into this. What is not known and not fully appreciated is that mycorrhizae will internalize other microorganisms from the soil. For example, bacteria will be internalized into mycorrhizal hyphae. So a mycorrhiza is not just a mycorrhiza. A mycorrhiza really is a mycorrhiza and containing a little community of other microbes. And even to the extent those bacteria, those other microbes go inside the hyphae of the mycorrhizal fungi. And there's an interaction there that is not explored yet, in that those microbes are interacting with the fungus, interacting with the bacteria. And what we think is happening is that some nutrients that are being produced inside the hyphae, perhaps nitrogen, that then is being passed on to the host plant that's really providing the carbon or the sugars to feed the mycorrhiza and then that whole system. So you see, it really is connecting the whole community of microorganisms to the plant. The soil microorganisms get connected to the plant via the mycorrhizae. So it's a really multi-partite. We used to think it's like a two-partite interaction with just fungus and plant. That's not correct. That's not correct. In fact, there are other microbes that are involved in that interaction, and that's an area that we're only now beginning to look at.
So that kind of brings me to the next exhibit, which is really more about the soil. And I'm going to ask you about some of the nutrients and uptake and things like that. But because we're talking about it, this rhizophagy cycle is something that has been really fascinating as I explore this. And I understood it to be that the root hairs themselves are taking in the bacteria and almost, I mean, I've heard some research almost farming the bacteria inside itself. And that sounds like what the mycorrhizae are also doing. Is that happening at both levels?
It's a little bit different with rhizophagy, and or we call it rhizophagy cycle, because it actually is a cyclic process. And what happens in the rhizophagy cycle, first of all, I'll say rhizo for root, right? Phagy for eating, right? So similar to mycorrhizae, right? Fungus root, but now we have rhizophagy, rhizo root, phagy eating. So root eating now. But what the way this system works is that plants will, grow their roots, put their roots down into the soil, and then they'll secrete exudates from the root tips. And these exudates are sugars, sometimes they're amino acids, and sometimes they're organic acids. Basically, what these exudates are attractants, microbial attractants. So they'll put this nutrients out in the soil, and they'll dissolve and go out away from the root, and these microbes will then follow that trail back, like Hansel and Gretel, back to where the idea, right? The breadcrumbs, the food crumbs. Okay, so they follow that to the root, and then they get to the root tip, and where they cultivate it. And then what happens is the plant will absorb those microbes at the root tip into the cells, into the root cells. So we know they absorb them into the cells because we could see them going in. We can see them crossing the cell wall, and the cell walls of the root cells at the root tip are really soft because they just divided and they're not hardened yet. Not like most plant cells more mature plant cells get hardened walls, but it's very soft at the growing root tips. So they take them in, these bacteria, mostly bacteria, but they also could be yeasts, and they also could be algae. We've recently seen that some algae are also absorbed into these root tips. And when they get absorbed into the root tips, what the plant does is it will then detect them there inside the cell. The plant cell itself, the root tip cell, detects the microbes in there and then secretes reactive oxygen onto them. Reactive oxygen like superoxide. Superoxide is just really potent form of reactive oxygen that will oxidize the cell walls off of these microbes and cause them to lose their cell walls, and then they'll lose their shape. They may be rods going in with their walls, but then they become these spherical protoplasts. And the plant continues, the root cell continues to hit them with this superoxide, and that not just the cell walls are oxidized off, but also nutrients are leached out of these microbial cells. So the process of getting nutrients from these soil microbes begins there. With microbes like algae, chlorella, for example, what happens is they get hit with superoxide, and then we can see the whole root tip turns green because of all the chlorophyll that goes into these root cells once they zap those algae with the superoxide. So then what happens is some of these bacteria, so they release their nutrients to the plant cells get them, and then the survivors in that process, and some of those microbes are fully degraded, some of them aren't. Some of them can survive that oxidation process. The ones that survive are then they go into the root hair initials, because don't forget at the root tip, you have cells dividing, that's where they go in, and then later on as the root continues to develop, those cells begin to mature. And one phase of maturity is when they begin to form root hairs, and they have these little root hair initials that look like little socks on these cells, just little elevations. And so the bacteria will go into those elevations, those little initials, root hair initials, and then they will actually produce hormones, plant hormones like ethylene and nitric oxide. And those happen to be growth hormones, and that will cause the root hair the root hair to begin to grow. Without any microbes in the root cells, you get no root hair elongation. The root hairs just don't grow. You get roots with no hairs on them. So those bacteria, those microbes, must be in those root cells in order to get root hairs to form. Okay, and then as those root hairs form, these microbes begin to secrete those hormones, causing elongation. And every elongation that happens, some of those microbes are shot out back into the soil, and from the tips of the root hairs. So, and then you have another growth spurt. In 30 minutes or so for the root hair growth spurt, the root hair grows a little bit, and then when it grows a little bit, then you have more ejection of bacteria, of microbes, bacteria, whatever the microbe is from the soil. You get them going back out of those tips of the hair. So you have growth spurt, ejection, growth spurt, ejection, growth spurt, ejection, growth spurt, ejection. So what happens is a few microbes go in at the root tip. Many copies of those microbes go out at the root hair tip. And these are the microbes that the plant can work with to get the nutrients. So essentially what the plant is doing is growing these microbes, or as you pointed out, as you mentioned, farming these microbes, getting a few, replicating them and putting them back out into the soil so they get more nutrients. And then later on, those same microbes will be attracted back to the growing root tip, go in and carry more nutrients. So it's a cycle. It just keeps going around like they keep going around. And so the plant gets continuous flow of soil nutrients by oxidizing these soil microbes, by bringing those microbes in and then oxidizing the nutrients out of them. Some of those nutrients are carried on the cell walls of microbes, and we can actually see when the plant oxidizes off the cell walls, we can see those cell walls being deposited inside the plant cell. So some nutrients, like there's some proteins in cell walls, there's some phosphorus in cell walls, there's metals hanging around cell walls. And basically what we have found out so far, what we've seen so far, is that every microbe carries a little bit different nutrients. So it makes sense that these fungi are taking in communities of microbes, now not just one, but different species, different strains, taking them in and then getting nutrients out of them. So it really is very much a plant farming these microbe situation. The plant is in control of the process. The plant is managing these microbes, attracting them, taking them in, getting nutrients out of them, and then putting them back out to the soil to get some more nutrients. It's very much like a farming process. It's really cool. And the plant is in charge. It's not the microbe that's in charge. The plant is in charge.
Thank you. That is absolutely fascinating. And, when you first start talking, it sounds initially like a predatory relationship, right? They're bringing them in, they're oxidizing them, they're zapping them. Only some are surviving. But then, as you said, they're spitting back out copies. Is there any data showing that the plant is selecting which survive and which don't, and so therefore which ones are getting shot back out into the soil?
Yes, there is this selection that happens. I'll give you a couple of examples. One is that, for example, if the plant needs nitrogen, what it will do is it will withdraw all nitrogen from the exudates, so that the only microbes that then can survive in that low nitrogen situation are the microbes that can actually fix nitrogen from the atmosphere. And in other words, make their own nitrogen. So the plant is selecting those nitrogen fixers. The other thing that's happening, again with regard to nitrogen, but it happens with other nutrients also. And but nitrogen is a good example. But another example with nitrogen is that the bacteria that are able to secrete nitrogen inside the plant are the ones that can survive the oxidation process with superoxide, because the forms of nitrogen, reduced forms of nitrogen, actually serve as protectants for the microbes inside. The more of the nitrogen that can be secreted by these microbes, the less damage the superoxide will do to the microbe. So when you have, for example, root hairs forming, the microbes that can survive in those root hairs are the ones that can make their nitrogen and then can secrete out that nitrogen as a defense against the plant superoxide, the plant produced superoxide, right? So that enables plants to keep processing the best microbes, the microbes that are going to provide nitrogen to it. And, so some of the microbes are likely fully degraded, and we can actually see them being fully degraded when they go in the root. So the ones that are not able to counteract superoxide using nitrogen or using some other kind of compounds that the microbes can produce to defend themselves, but nitrogen is the most likely one because it's a primary metabolite, and it's one of the first metabolites produced. So some other antioxidants or secondary metabolites, and it takes a while. But what happens is inside the root hairs, as they elongate, there's no time for secondary metabolite production. So the antioxidants have to be, something that can be produced very fast. Things like ethylene and nitrogen and ammonium nitric oxide, chemicals like that protect the microbe.
Could you see mine? Mine was going in and out. Are you good? You can see me, hear me?
Yes, we can.
You're freezing.
Hold on 1 second. I'm going to plug my phone in.
You're freezing up.
This is fascinating. Wow. I never thought that it's amazing. It's really, really amazing. So much detail.
It's crazy, isn't it? Seems
crazy. Yeah, it is. It is.
It's a different view of how plants work, a different view. They absorb microbes and get nutrients out of them. April, you're still frozen.
I know. Sometimes the internet went down. I love the mug. I was looking at the mug too that you have with your grandfather, with your grandkids, I assume. Your mug.
Yeah. Oh, yeah, that's right. These are grandkids. Yeah, those are grandkids did that. You see that?
So cute. Yeah. What a talent. What a talent.
Really do. She's like 5, this one who did this, and her sister is like 3 now. So there's two granddaughters. Amaranth is one, and then River Willow is the other one.
Beautiful names.
Plant names, right? They're plant.
I love them. That's so cute. But what a talent. I was just looking at it. I was paying attention to you, but then I noticed, I was like, that is really cute. I love it.
Yeah, and my daughter is so good too. See, my daughter did that.
Oh.
Yeah.
Wow.
Well, you guys didn't know, but I'm a mycologist, so so the fungi have a special place, right, in my lab, and so those questions about fungi are no problem at all.
Oh, that was wonderful. So let me just send her a little. I think that she's just trying to connect again. You know how it is, and sometimes with the internet, but hopefully if we can take a couple more minutes of you, but if you have to go, we completely understand as well.
Well, you can take a few more minutes. Hopefully she
Thank you.
Connect. Yeah.
Yeah, I think probably she's just moving around the area. So cute. So I'm sure so I can say that your daughter is actually, very fascinated, with the nature too, with the everything.
She is fascinated by it. She's really busy right now, but whenever we visit, we like to go and do some in the summer, at least, and fall, do some mushroom hunting.
Oh, that is amazing.
Yeah, and now my granddaughter Amaranth, she's getting into it too. But she's only 5, so but she started, she knows how to well, she doesn't have to do very much to get into her grandfather's heart.
Oh, I'm sure. I'm
That's
sure
not
it
that
was
hard.
just very easy.
Yeah, yeah, it's very easy. Exactly,
Well,
exactly.
do you know how they are? They're amazing. They're
Yeah.
really cute.
Oh,
That's
yeah.
wonderful. Actually, April asking me, sending me a text that she's having problems, the service problem, but she's saying if you can ask him about to explain the role of NPK. So I think it's nitrogen, phosphorus, and yeah.
Yeah. Okay. So rhizophagy cycle actually brings more than NPK. This is nitrogen, phosphorus, potassium. These are 3 very big, important nutrients. So they're carried in rhizophagy cycle to the plant. The microbes take there's 2 ways that nutrients are provided to the plant. One way is that the microbes in the soil will solubilize nutrients in the soil, right? They'll become soluble, they'll go into the soil water, and then the plant can just absorb it into the roots. Okay, so that's happening. We know that's happening. We've always known that's happening. I mean, for hundreds of years, we've known that's happening. In terms of rhizophagy, okay, this is actually a little bit different process in that the plant root cells are internalizing those microbes, and then they're getting the nutrients out of the microbes by oxidation, right? This oxidation process. And so we can actually do experiments to separate out which nutrients come from solubilizing in the soil by the microbes versus those that actually are oxidized out of the microbes, out of the bacteria and other microbes that are internalized into root cells. And when we do that, all the nutrients are involved, but very, very important, it appears, for the oxidation rhizophagy cycle process is divalent metals, things like manganese and magnesium and iron and zinc and those things. And those, it appears that the microbes themselves carry that into and some of those are very hard to get otherwise. So because the plant internalizes the microbes and then forcibly takes the nutrients from the microbes, it's able to get those nutrients. So plants, and then the other way, if we go back, we have the mycorrhizae now. The mycorrhizae are the third way that plants can get nutrients from soils. So you have your solubilization in the soil, and then the plant just absorbs from the soil water, and then you have your rhizophagy internalizing, engulfing these microbes, eating soil microbes, and then forcing nutrients out of them, and then you have your mycorrhizae. So those are the 3 main ways that nutrients can be obtained from microbes, basically. Yeah.
That's amazing. Oh, she's back. Okay.
Hi, I'm so sorry. I don't know what happened. The mystery, everything was fine, and then it stopped allowing me to connect via my computer. So apologies. So, Dr. White, I know this is probably beneath your pay grade, but one of the things that we're trying to explain to people is some of the roles that the nutrients play in plant development and plant growth. I know for cannabis, NPK are always the 3 winners, but trace elements are also required. Can you just give an overview of NPK and its role in plant development?
Yeah. Okay. Nitrogen, phosphorus, potassium. Okay, these are key nutrients for the plant, right? They need all the cells. Nitrogen and phosphorus and potassium are key components of cells. So parts of plant cells are made up of these nutrients. But we also have all these micronutrients and other macronutrients too, but they're all important. And so you can't, even if you had NPK, if you don't have these other soil nutrients, plants are not going to be able to thrive. They're not going to be able to grow. They will not. You have to have a whole broad base of nutrients. NPK. The problem with just applying NPK is that if you apply too much, you suppress the rhizophagy cycle. You suppress rhizophagy. You suppress, especially with nitrogen, you suppress the capacity of the plant to get nutrients from soil microbes. They just in general, over-fertilized plants don't do rhizophagy, or they minimally do rhizophagy. They tend to lose nutrients when they're lose microbes. I'm sorry. They tend to lose microbes when they're over-fertilized. And so essentially putting too much NPK on causes plants to lose their ability to grow and get nutrients out of soil microbes. So basically you're messing up the process. I collaborate with a corn breeder, Walter Goldstein, in Wisconsin at the Mondawmin Institute. But Walter grows organic corn, and organic corn, more than organic corn, nitrogen fixing corn. So what he is doing, he's taken these land races from South America and Mexico that are highly nitrogen efficient and don't need any kind of nitrogen fertilizer on them, or very, very minimal fertilizer, right? He's taking these and then trying to make cultivars out of them so that then organic growers can use it without having to put fertilizers on. What Walter has found is that corn plants that are fed this NPK, right, too much fertilizer, a lot of NPK, a lot of our conventional corn varieties, their roots will go down far into the soil. They'll follow where the fertilizer goes. They'll follow the fertilizer down, and they also don't branch very much. You have very little branching. Instead, they tend to be spindly roots. But the ones where that aren't fertilized, they will actually form in a layer on the surface, near the surface of the soil, just beneath the surface, and abroad with lots of branching happening in there, with lots of microbes. They take in lots of microbes, whereas we don't see very many microbes on those NPK fertilized plants. So it is really overuse of NPK. Yeah, we can push crops using NPK, but we also take away the ability of plants to get their own nutrients using soil microbes, using soil microbiology. So it's a problem. And then you have, if a plant is doing rhizophagy cycle by absorbing microbes, getting their nutrients, the plant can regulate that process. It can attract microbes that it needs to get nutrients it needs. But if you're putting fertilizer on, that process is inhibited, and instead plants will just take in the NPK. But what that means is, well, they're not getting all these, these metals, these divalent metals, these micronutrients, the calcium and the iron and the manganese and the magnesium. They're not getting it. So then you have issues with those nutrients. So basically, too much fertilizer, we take away the plant's ability to take care of itself, and then we put that all over on us. And the problem is that we don't have perfect knowledge about what the plant needs. The other issue is, for example, when we begin to lose our microbes in plants, the microbes that go on roots and in roots, and they actually will go all over into leaves and everywhere in the plant, in the fruits. When we take away those microbes by over-fertilizing or sterilizing seeds or doing something like that to kill microbes on the plant, what we do is we make plants more vulnerable. One of the things is they're more vulnerable to diseases, because the microbes in plants will also suppress fungal diseases in the plants. And that happens because the bacteria in the plant will colonize pathogenic fungi and make them non-pathogenic. They will make them avirulent or non-virulent so they don't cause disease. I mean, you could consider that, and I think you could think the same thing about the human gut. The microbes are part of our immune system, right? So the same thing is for plants. The microbes are part of the plant's immune system, and without them, what happens is plants are susceptible to all kinds of fungi. Fusarium is an example that will just eat the plants up. The resistance to these pathogens breaks down. So, and this is one of the outcomes of our modern chemical approach to agriculture. The other issue, and we haven't talked about that, is the microbes. They're taken in at the roots, but they don't stay in the roots. They also go all over the plant into the leaves. And one place we see them are on the trichomes, in the trichomes. So just to give you an example, with cannabis, we have trichomes, glandular trichomes and filamentous trichomes all over the cannabis, on the developing fruits and flowers, and on leaves, and on stems, especially in early time of development, but mostly in those fruits, right, flowers, inflorescences. Those trichomes are actually very early on, they're filled with microbes that appear to be fixing nitrogen based on our histochemical staining. We do a lot of microscopy work, and so we developed some stains, microscopy stains, for visualizing nitrogen and nitrogenous compounds and some other compounds in there. And so these, particularly the glandular trichomes in cannabis, become filled with nitrogen because of the interactions with the microbes inside those trichomes. And so we could actually see, early developing glandular trichomes have just a few microbes in them. The plant uses superoxide again to remove the cell walls, and then it replicates those microbes. And then the microbes inside the glandular trichome, inside the plant's hair, glandular hair, right, glandular trichome, will begin to secrete nitrogen. And we can see that. Hops is another plant in the cannabis family, right? Cannabinaceae, where we see the same thing. A lot of nitrogen gets produced inside these glandular trichomes, and we know that many of the chemicals, of the terpenoids, the cannabinoids, right, that form in those glandular trichomes, we know from people who've done experiments that they have antioxidant properties. And people have used, they've thought, well, this means we can use it as a medicine, as an antioxidant, right? But inside the glandular trichome, what we think is actually going on with these terpenoids and cannabinoids is that they are, again, antioxidants. But what they do is they will scavenge oxygen from the air and enable the microbes to fix nitrogen more efficiently. Because what happens during nitrogen fixation is that if there's oxygen present, the microbes cannot fix nitrogen from the air. So the nitrogen has to be limited. And those cannabinoids and terpenoids inside those glandular trichomes are we think their role there, at least their initial role, we think, is to limit the amount of oxygen around those bacteria so that they can more efficiently fix nitrogen from the air. So there's a little bit of chemistry going on there, and it's almost as if you could consider a glandular trichome to be a sort of precursor to the nodules of legumes. Legumes have these root structures called nodules, right? Plants in the bean family and so forth. They produce these nodules. These nodules actually form from root hairs from another kind of plant hair, but these plant hairs are all similar, in some respect. Root hairs are similar to trichomes and so forth. They're a little different structurally, but essentially they're the same thing. And they basically are microbe processing structures. And this is what we're finding.
So are those microbes that are in the glandular trichomes, those have made their way up the plant from the root hairs, from absorption into the root tips?
They have. They come from the microbes that the plant absorbs in. Now, a little bit of complexity here is that some of these microbes are carried in seeds, right? But the plant essentially are taking these seed microbes because they're really good. They're already good. They're really good microbes. The plant takes the best of the microbes from the root cycle, from the soil, and then puts it up in the seeds. Okay, so when the seed germinates, that baby seed already has the microbes in it that it needs to survive. So it's kind of like the lunchbox. The seed. You could view the seed as people used to say. What is it? It's in the baby plant in a bag with some food. It's lunch, right? In a bag with its lunch, or in a box with its lunch. Okay, but now, because those microbes are going too, it's kind of a baby plant. The seed is a baby plant in a box with its lunch and microbes to get some new nutrients, for it from the soil. The other issue is that these microbes are important for plant development. And if we take all the microbes off, plants won't develop properly. They just won't grow. They need those microbes in their cells and in their tissues in order to grow appropriately. They simply won't grow without the microbes.
In hearing you talk, I hear all the time, or I have heard, I don't know who said it or if anyone said it this way, but that we know more about the surface of the moon and the bottom of the ocean than we do about the inch beneath our feet. And just listening to you and learning what I've learned in researching this exhibition, the approach we take to cultivation seems epically clumsy, in terms of soaking seeds in hydrogen peroxide, cranking things full of nutrients. And is the thought behind some of your research that if we understood these processes more effectively and robustly and thoroughly, that we could drastically reduce the amount of fertilizers that we're putting into soils and plant growth and feeding ourselves?
That's exactly what I think. I think that we went down the chemical path when we became able to produce a lot of fertilizer, right? A lot of nitrogen chemical fertilizer, and that happened, just before the Second World War or something like that. When we figured out the Haber-Bosch mechanism, the German chemical, when they started creating nitrogen, right? Learned how to create nitrogen, and that's when we really started doing that. But we believed that that's how plants work. We believed from way back, from the 1800s, way back, we believed Justin von Liebig, right? That they needed chemicals, nutrients. That's the way they work. We thought that. That kept going back, and that went back, and it kept going up until very recently, until we figured out that that's not really how plants work. At least, I mean, this is what I say. This is what we say in my lab, and what other people are saying is that plants are much more microbiological than chemical, and that if we, I would say another thing about that. No one has been any malicious, intent to sell chemicals or anything like that. We go down this road, because with the best of our knowledge, right? And we thought chemistry was it, right? We thought that was it. We didn't know that we're going to create all these problems that we have now, and it just so happens that no one could see these microbes being absorbed into plants. They couldn't see them going into the plant roots. They couldn't see. It really wasn't until people began to study endophytes that they could then say, well, something else is happening here, right? So, and I'll say another piece about that. There have always been people who didn't feel right about putting all the chemicals on their soils and on their plants. It was kind of an intuition, right? They didn't know why. They understood that it didn't seem right, and they didn't know exactly why that wasn't right. They didn't know anything about plants absorbing soil microbes into their cells, but yet they kept growing plants, crops organically. They had an intuition about it, I think it was an intuition. And I think that the reason that rhizophagy cycle has been so well received is because those people, and there's a lot of people, were waiting for it. They needed that information to confirm what they already intuited, right? What they already felt and what they already, we don't know the biology of it, all the biology. When you think about, soil chemistry and soil biology, it's very complicated, right? But rhizophagy cycle, from the bare outlines of it is not complicated. It's not that complicated. And people can understand it. They can see it. They see how it works, and they have, it confirms to some extent what they already felt, that the chemicals is not the best way necessarily to grow crops. In fact, it really is. It's one of the worst ways because of all the damage we do to the environment, and we basically paralyze the plant's capacity to do it. And the other thing that happens is because plants are taking in microbes and interacting with them inside their cells, the plants themselves get nutrients, that's for certain, but because it's oxidative interaction, the plants upregulate oxidative stress genes, antioxidants, and things, phenolics, and things that protect them from reactive oxygen. And that means their own cells are protected from reactive oxygen, but that means they're also protected from stress, because stress is essentially oxidative stress, and that means reactive oxygen inside the tissues. So plants doing rhizophagy cycle are more resistant to stress, like high salt or like heat or any stressful condition. Heavy metals could be converted to stress. Any harsh treatment, if you're growing cannabis, if your plants dry out and you have more microbes there, you're growing them organically, then they will be more resistant and more likely to survive than plants that you're just growing by putting a lot of chemical fertilizer on. So so anyways, there's lots and lots of benefits because if you permit plants to do rhizophagy cycle and encourage that soil biology, use cover crops and stuff like that if you're growing field crops. So there's lots of reasons that the microbes are better than the chemistry. Now that doesn't mean we don't do chemistry, but maybe we use the chemistry in a much more restrained way, a much more limited way. We use chemicals, we use the chemistry like biostimulants, at lower levels, 20% of what we use now, something like that, just enough to push the plants like that, just a little bit. Use other organics, use other microbes in there, use algae or algal extracts, things like that, to, that where you don't have to use and the plants like I said, we know when we feed them, if we feed plants chlorella, which is green alga, they take them right in and get the chlorophyll out. So a lot of these algal extracts are chlorophyll. They like that.
And that these plants that are grown in this way, you're saying they have these more antioxidant properties that translates into a better product for human consumption as well, right? I'm imagining that those antioxidant properties, we then benefit further from that increased, potential within the plant.
That's absolutely correct. There's a lot of people who are working on nutrient density, right? These phenolics and food quality, right? Working on food quality. As it turns out, that's an endophyte property. That endophytes, plants with endophytes, with doing rhizophagy cycle and getting other endophytes into their tissues, are producing these phenolics and other kinds and even enzymatic antioxidants, superoxide dismutase and vitamin C, ascorbic acid, and stuff like that. So all of that is an outcome that increases in plants when they have their endophytes, when they're doing rhizophagy cycle. And yes, it is food quality. There's the idea, there's data that shows, as reported, that since around the 1950s, about the time we started doing chemical fertilizers, that food quality has been declining. Now, it's not clear that it's only, the endophyte issue here with our foods that we're suppressing that, but to me, it's a likelihood. It's a correlation, almost an exact correlation with the beginnings of chemical fertilizers, and it's the flip side is it's easy to grow things, to grow crops by squirting chemicals on them. It's expensive though. It's expensive, and it's not sustainable. We're wrecking the earth, we're wrecking our health. All kinds of it's not sustainable. It's not sustainable, but it's easy, and we can do a lot of land like that. The flip side is that if you try to grow crops biologically, it takes more work. You're putting cover crops on, you're using biostimulants, maybe you're adding microbes to the soil, you're doing things that take more work, right? You're more involved. So if we were ever to change our agriculture on the planet to do more biological agriculture, we're going to need more farmers, not fewer farmers. Right now we're in a trend where you get fewer and fewer farmers, and they're all doing lots of acreage and squirting lots of chemistry out. Well, we'd have to change that. We're going to have many, many more farmers with smaller land, putting the work in to grow crops biologically. But the benefit is they're going to be good. It's going to be good crops. It's going to be good food for people. They're going to be rich again.
And I would love to see that. I have a young son, and I have made farming a part of our, what we learn on a daily basis. And I see a lot of parents and people trending toward that. So I hope that we do see a trend toward smaller farms and people learning this more as a mandate in schools rather than just, a hobby. So I have one more question for you, because I'm cognizant of your time. I know I wanted you to get back to your research. We need it, but this is a cannabis exhibition, and we all know that cannabis has this unique property, these psycho and these psychoactive effects. And one of the things that I've noted in this exploration is all of these very intricate dances that take place at the various levels between microbes and the plant, between the cannabinoids themselves and your endocannabinoid system. And do you ever ponder these things more fundamentally, like then what's going on in the microbe? What is the microbe farming itself? Are there any sort of more philosophical ways that you think about this exchange and these interactions when you're, in your own time?
Well, there are some things that we see, one thing about scientists is we tend to be more constrained, right? So we tend to be more conservative in our interpretations. But a couple of things that amaze me. First of all, the beauty of the process, the rhizophagy cycle, is beautiful, that it's a cycle. That is beautiful. That really is, that the plants are able to do this. Another thing that is really interesting is that there is this interaction with the plant, and in which some of these microbes in the meristematic areas of the plant actually get incorporated in, because they're going into the dividing cells, the meristematic cells, right? And when the meristem divides, when those cells divide, the nucleus pulls apart, right? So then the chromosomes divide, right? So there's no nuclear envelope, right? But they go in when there's no envelope around the nucleus, and then the envelope reforms around the nucleus, and bacteria get trapped in that nuclear envelope around the nucleus. So there is the possibility that the presence of the microbe there alters gene expression in the plant. So you have the possibility is that, not only is the plant affecting the microbe by getting nutrients from it, but because the microbes are in so near the nucleus, that also may be turning genes on in the plant. And this, I go back to my friend and collaborator Walter Goldstein. Walter tells me when we put these microbes, these nitrogen fixers, in his corn, what he sees is these plants appear to be evolving more readily in his nurseries where he's growing the corn, right? Breeders have these breeding nurseries where and he can see the changes happening in the corn. And the possibility is the other thing that he shows. I mean, it's really interesting. When they try to put genes to limit crossing into the corn, because this is what breeders do. They don't want, for example, they don't want these corn to get GMO genes in them, right, from other corn. So they try to put these GMO restricted these so that it won't outcross, right? They just want in-crossing so they don't get that. The problem is they keep getting rejected by the plant. They put the genes in, they see them there initially, and then in a year or two, boom, they're gone, like that. They're gone. They're silenced. So the gene is being silenced, or the genes to limit outcrossing are being ejected. So the plants, it's almost as if these corn plants with their nitrogen-fixing endophytes that go into their nuclear envelopes, it's almost as if these plants want to adapt or evolve, right? Want to adapt or evolve. They kick out these hybridization, these crossing limiting genes. It's fantastic. So it is, what really does amaze me is a couple of things more, that we missed all this, that there's a world there that we don't understand. There's a world of activity that's happening that we don't understand. And, this is, for somebody who studies endophytes, this is really important, because it means, wow, endophytes are more and more interesting. And of course, the possibility is we can make, cultivars, new cultivars of plants that are rich in endophytes. And, it's I mean, those are the big things that I think about.
It's mind-boggling. It really is. And I love to think of the plant, determining its own future, despite all of our efforts. Otherwise.
Yeah.
It's funny, one thing that I'm from Colombia, and like every single time that I go to Colombia and I eat the regular food that I eat from here, it's a huge difference the way that I feel. So everything that you're saying is completely true. Like, it's just, yeah.
I believe you.
Well, Dr. White, I am so grateful for your time. It was absolutely astounding to me to get you, and to be able to jump on with us is really, we're so grateful. I would love to include some of the graphics from some of your presentations, if you're okay with that, as we play this back. So, okay, perfect. And I'll be in touch. I would love to, like I said, I'd like to follow up with you every now and again and keep abreast of what you're doing, because it's fascinating.
Certainly, April. And Gloria, it was nice to meet you too.
Thank you so much. Very nice to meet you too. And thank you for sharing the stories about your granddaughters. They're very beautiful and talented.
Thank you so much.
Have a great weekend.
Okay, you too. Both of you too. Bye-bye.
Thank you. Bye-bye.